Well, good day, everybody, and welcome to our lab for week 4. And this week, we're going to concentrate on the medial surface of the brain, the so-called mid-sagittal section of the human brain and brainstem. And we'll look at the gyri and sulci. We'll also start segueing into the brainstem, which we're going to be doing next week, or beginning to do next week. And so, largely, the sheets that I provide to you are guides. And I want to also explain to you that I reserve the right to hold you accountable for the lab material, the identification of the structures on any of the lab presentations like this that I do. So week 4, again, medial and basal surface of the brain. The basal surface is a basal view, is the brain upside down and looking at it from the bottom. So let's start out. These are all from the Haynes Atlas, the Duane Haynes Atlas. It's a classic, as I mentioned. It's on the reading list in the syllabus, and these are right out of it. And you can use any edition you want. You can get one for 50 cents. That's 5 editions old. These are the same pictures he probably took around 1970. Dr. Haynes is still out there. He's an emeritus professor somewhere. I forgot where he was. He is now. He's the most well-known medical neuroanatomy educator of all time. And so anyhow, I just want to tell you where I get these pictures so you can go and get them. You have them here. And essentially everything on here is clear cut. If you have questions, you can go look it up online. But for the most part, you know me, I told you, I'm a very straight shooter. It is what it is. I won't go out of my way and say, for instance, on a quiz and go, tonsils of the cerebellum, and I didn't even mention it. We're going to mention the cerebellum. Heck, we're going to cover the cerebellum at length. But I won't take things out of context. This week is medial surface of the brain, and you'll see. So I think we ought to start off with the gyri and sulci of the brain that we didn't do last week. We just did a lateral projection last week. Also, these videos that I made, that I posted, those were made, I'll be very honest with you, I flew out there and it was right before Thanksgiving, and I flew out there to Dallas to do those, and those were done on the fly. That production team set up the lighting. There were specimens there. I didn't have much to do with the specimens. We have a pretty decent collection actually there at Parker. So I took the best that I had at the time, and I went off the top of my head. So the zoom on those isn't quite on some of the structures the way we want. I'm going to come back and redo those this spring or summer, and probably in the spring. And the production team's in the process of labeling them. I told them, go ahead and give them to me for my students, because they don't need the labels. They got this, and it'll help them learn by labeling it themselves. So we'll start out by probably one of the largest and most important gyri on the medial surface, and that is the cingulate gyrus. Let's put a mark by these for sure. I'm going to probably ask you, cingulate gyrus. And the cingulate gyrus literally runs the full length of the corpus callosum. Now the cingulate gyrus, we talked about that in the cingulum in our subcortical fiber lecture this week. If you didn't watch that, please do. And it's got a lot of anterior connections into the frontal lobe and has a lot to do with neurodevelopmental abnormalities and disorders. Well, the corpus callosum we talked about quite a bit already. And of course we have the rostrum of the corpus callosum. You see here the rostrum of the corpus callosum, which is here. The genu of the corpus callosum, which is here. The body of the corpus callosum, I'll put dots on it, which goes back to here. And of course, the splenium of the corpus callosum is this big rounded area back here. Again, the largest commissural or side-to-side connection axons in the human nervous system. Now, above the corpus callosum, right before we get to what's called the cingulate gyrus, we have a sulcus called callosal sulcus. Now that makes total sense, doesn't it? That callosal sulcus is sometimes called the sulcus of the corpus callosum. I'll take either one. Okay. The next most important structures that we see on the medial surface of the brain, probably, I think everybody would agree, is the paracentral lobule. And Dr. Haynes is fairly traditional, but he does this a little bit different. He has anterior and posterior paracentral gyrus, and you can see both kind of right about here. These together are called, and I'll go ahead and write it down for you, the paracentral lobule. paracentral, sorry, lobule. Not a lobe, but not a gyrus. It's two kind of areas. And this is kind of an interesting area, and it's sort of famous because on the outside edge, the lateral view or projection of the brain, we have the central sulcus, which we looked at last week. And the precentral gyrus is right terminating here, and the postcentral gyrus is here. These two guys kind of blend up here. So there's a lot of neurons that are doing some duality of function, especially in the lower limb. So that's kind of well known for that. Not that big of a deal, but it is very, very commonly noted, so we'll know it as well. The next structure is the marginal sulcus. And the marginal sulcus that you see labeled here is actually the borderline, if you will, right here, separating the paracentral lobule, okay? Next thing on tap is parieto-occipital sulcus. Parieto-occipital sulcus, very easy. Why? The parietal lobe, we talked about that in our lecture this week on the cerebrum, and the parietal lobe, and down here is the occipital lobe. So that makes total sense for nomenclature, parieto-occipital sulcus. It's beautiful when a plan comes together like this in neuroanatomy nomenclature. It doesn't always, like the next one. The next major one is a little bit more of a horizontal sulcus, right in here, and it's called the calcarine sulcus, C-A-L-C-A-R-I-N-E, calcarine or calcarine, some people call it, most call it calcarine. Now the calcarine sulcus is a bit more horizontally oriented, and it essentially is a major division, between the superior aspect of the occipital lobe and the inferior aspect. So what's our occipital lobe? What's it famous for? It's our primary visual cortex. Brodmann area 17 back there. So around this area is where visual imagery neuronally takes place. Not interpretation, but imagery. And that's just called the pericalcarine region, right? You'll hear that or read that in a book. Now above it is a gyrus, a region of gyrus called the cuneus, right there, the cuneus. And then below it, it's called the lingual gyrus. See that? Lingual, like a tongue. Somebody thought that this lower portion of the inferior aspect of the parietal lobe is the lingual gyrus. Regardless, these are all right around in this area as target shape for visual imagery, for neuronal firing due to visual stimuli from the retina, from the system. Okay, so a very, very important area. Let's see, what else have I forgotten? Anything there? One other area probably on my list, I think it's on my list, and that's precuneus. So if this is cuneus, you know, cuneus is posterior to the parieto-occipital sulcus. Precuneus would mean in front, and that's part of our occipital. That's part of our parietal lobe visual association center. That would be Brodmann areas 18, 19, around in there. So like face recognition and things like that is precuneus. As far as gyri and sulci, I believe that covers our basis on the medial surface. Now, these other structures, I think you probably know from our lectures. Again, the lab and lecture in here should really not be very different at all. So, for instance, you know that this is the mesencephalon, or the midbrain. And then here's the pons. I'll put a P on it. There's the border of it. And the medulla is down here. I've just put an M there. I want you to recognize the ventricular systems, and I point that out in the video as well. You can see this structure called the fornix right here. And the fornix is sort of the bottom of this septum pellucidum. It just says septum here, but it's septum pellucidum. And that's that divider, that tissue, that septal tissue that divides the right and left lateral ventricles. There's a little aperture here where cerebrospinal fluid can come out into the third ventricle. And I believe our next pictures will have some of that in it as well. So let's move on and see if I'm correct. So this is basically the same brain, or a similar brain, with the brainstem removed. So let's take a look at that and be able to recognize these structures not only on the Haynes Atlas photos that I have here in these presentations each week, but also in drawings and even lecture materials. Because when I provide the images for the exam will be online, and it's just going to have a picture like this, and it'll say, you know, where the X is, which structure is that, A, B, C, D, pick it out below. So multiple choice, pretty straightforward stuff. Might have some correlation questions, but not much. This is mostly idea. Correlation, I have you for Got you for lecture questions, so we don't have to do too much of that. So this particular projection, again, is a mid-sagittal, and it just has the brainstem removed. And I'm trying to make sure that everything that I mentioned before, there's nothing really new here. So the sulcus of the corpus callosum you see here, again, that's sometimes referred to as the colossal sulcus, and it separates the corpus callosum from the cingulate gyrus. The cingulate sulcus is above the cingulate gyrus. So that's the cingulate gyrus. There's the cingulate sulcus right here. Where is it? There it is, and cingulate sulcus. And then you just follow that back with your probe and lab, just go along. And all of a sudden it turns up and goes whoop, and you can go out of the brain, right? It's like a maze. That's the marginal sulcus. The marginal sulcus, and of course, who's in front of it? Paracentral lobule. Paracentral lobule. Okay? The rest we already did. Anything else on here I need to elucidate? Not necessarily, I think, for our purposes right now. There is some cool choroid plexus laying right in here. There's a little bit of the uncus. We talked about that, the uncus of the temporal lobe, the uncinate fasciculus and things of that nature. We'll come back and visit that in a little while when we look at the basal view of the brain. So far this is basically the same view, isn't it? And so repetition is beautiful. So you know those structures I just labeled on there will be on your picture exam. So no surprises. The dish doesn't do surprises. It's all explained ahead of time. There's no surprises. What's fear? What's anxiety? Anxiety is fear of the unknown. So people have test anxiety because they don't know, oh, you know what, it's going to be killer. Nah, it's going to be what I tell you. I don't lie to you. So here's a lateral view of the brain, a lateral projection. And this is pretty cool because we took a blade, Dr. Haynes did, and he dug this out. So he kind of dug out some of the, kind of on the inferior frontal region. He went into the parietal lobe and down in the temporal lobe. And what's this out in here? This is the insula. Remember the insula from lecture? And you all shake your head of yes, of course. So there is the circular sulcus. And so the circular sulcus it's got to be labeled on there somewhere. I'm not even seeing it. Yeah, I'm sure it is. Well, they have short gyri, gyri brevis. Dr. Haynes is Latin, is a gyri longus and brevis, right? They're called short gyri up front. And Anglican, English non-Latin, they're called the long gyri. Gyri longi. It makes you sound smarter when you're doing like a Latinus, you know. Gyri brevis, brevis, plural. The central sulcus of the insula, not much else there to show you, but I wanted to show you that because that's fairly unique. And that insula is a very crude, primitive sort of mini brain within the brain, just next to the basal ganglia. So it's got some motor control function. It's got some alertness function in the human brain as well, kind of keeping our brain online even as we sleep. And the attentiveness, if you will, the readiness of the central nervous system or the brain. So that leaves us with what we want to finish up with this week in the brain, and then we'll move on to brainstem and cranial nerves over the next 2 to 3 weeks as we study the brainstem and cranial nerves in lecture starting in week 5. And so the basal view is just this brain turned upside down, and this has had the brainstem transected or cut across. And this is at the level of what? So the brainstem is composed of 3 areas: midbrain, pons, and medulla. And this is cut. You see Minnie Mouse, see the ears and her headband there, substantia nigra? That's midbrain, mesencephalon. And her little nose, her mouse nose, her little ratty nose there is a cerebral aqueduct. Look how tiny that is. Okay, what's this X? Anybody know what that X is there? We call this the optic complex sometimes. Optic nerves, you see it labeled there. Optic tracts back here, and the chiasm. So the tracts go back to the thalamus and then goes through our visual pathway all the way back to the occipital lobe, to the very posterior aspect of it, back by the cingulate, the calcarine sulcus, the pericalcarine area for visual imagery production. Some other things, septal areas, just this inferior portion of the frontal lobe. Gyrus rectus is the straight gyrus. That's important. That's the only straight gyrus in the human brain. Gyrus rectus. the olfactory bulbs and tract. I think we'll save the cranial nerves for next 2 weeks lab, so we won't label those today as far as responsibility, but ultimately we're going to know them anyway. So what do I want you to know about gyri and sulci? The gyri and sulci on the inferior view of the brain, one of the most famous is called the parahippocampal gyrus. You see that over there? Parahippocampal. Para means around, and it means it's around the hippocampus. So if we were to take a blade, a scalpel, and just cut through here, down the whole length, and here is the length of this parahippocampal gyrus. Runs about like that, and it's doing it over here on that side. So they're the two linear gyri, if you will, that kind of hugs the brainstem. They're most medial or most intimate to the brainstem. And so the parahippocampal gyrus, know that one, that's very famous as well. The uncus, I mentioned, the uncinate fasciculus, and the tentorium is a dural sheet in which the brain sits on, literally. The edge of this temporal lobe, very anteriorly, is called the uncus. And that's where that uncinate fasciculus connects into that inferior frontal region. This is famous for herniation. So if somebody comes into the hospital, they go to the ER, they've been hit in the head or they've been in a car accident, and their intracranial pressure is through the roof from a bleed or whatever, and they're going to have pupillary, they won't have perla, their pupils won't be equal and reactive to light and accommodation. That's a sign you got increased intracranial pressure. You look on fundoscopic exam, when you look in the retina, you're going to see papilledema, the bulging optic nerve disc and cup, and you may see some hemorrhage in there, but you'll also have headache. The patient will have headache, projectile vomiting usually. Why am I telling you this? Increased intracranial pressure. If they go in and say, well, let's see what's going on, and they think you have meningitis, trauma, they would obviously figure it was probably traumatically induced as an etiology. But if you just went to the hospital that sick, and people have had spinal taps done to look at, that's how you get CSF, because conus medullaris stops at it depends, L2-3, L1-2, right, in adults. So if you go down to L4 in an adult, you're not going to hit the conus. You stick a needle in there, and the cauda equina will move out of the way a little bit, and it's pretty safe. It doesn't feel great, but it's pretty safe. You pull out CSF, you can see if there's red blood in it from hemorrhages, or you can see if there's proteins in it from myelin breakdown with MS. So that's when they do that. If the pressure's too high, it'll shoot out of the needle across the room just about, and everything gets the cerebellar tonsils, which are against the foramen magnum. They can get sucked down in there, but this guy, the uncus, against the tentorium, can get sucked down, and that is not a good thing to do, and it's not uncommon. But the uncus is famous because of that uncinate fasciculus, as we discussed. So those are really the parahippocampal gyrus is really the dominant feature, if you will, on the inferior surface. Now remember, we did the 3-finger rule on the lateral surface, right? Superior, middle, and inferior temporal gyrus, and a 3-finger rule up here, superior, middle, and inferior frontal gyrus. Well, you can see the inferior temporal gyrus pretty good on this basal view. It's kind of hard to show it in the lab videos unless you turn it upside down. By the way, in the lab videos, when I say this is the right side of the brain and you're like, no, it's not, it's because they flipped the camera. The way they had it set up, it wasn't over my shoulder, it was in front of me. And once we started, I said to the guys during the break, I'm like, we can't learn this upside down. They said, we're just going to flip it. So right, if I say right or left, I'm not wrong, it's been flipped, much like your visual system does, cortically. What else as far as gyri and sulci? Orbital gyri. Orbital gyri. Really simple. These little gyri up here, they're depressed. It's hard to see on just a picture, but they have a little concavity, and that's where the orbit of the skull is for your eyeball to be in. And I think for now, I think occipital gyri is pretty self-explanatory. So I wanted you to see that parahippocampal gyrus and that anterior aspect of the temporal lobe. This last image is virtually the same It was probably the same brain, in fact it is, before he transected the brainstem with a scalpel at the level of the mesencephalon or the midbrain. So that's what you're looking at there, is a basal view, an inferior view some people call it, or a basal view of the brain. And there's the big pons. The pons means bridge, so it's like a big kind of shield, like really unique looking. And of course the medulla oblongata is right here, and it looks very columnar, and those are called the pyramids of the medulla, which is the pyramidal tracts, the motor tracts. So your motor system decussates right there. Okay, so all these structures stay the same. Parahippocampal gyrus right there, uncus. What else that we talked about? I think inferior, probably the inferior temporal gyrus right in this region. So this is actually kind of the start of our cranial nerve and brainstem segue, and you can see the midbrain from the basal view from the front, and that's the front view of the brainstem. All you can see is what's called crus cerebri, sometimes called the cerebral peduncle. Crus cerebri. It's a crossing of the cerebrum. Then you see the pons, and this is called the basilar pons in the front, and I have videos for all this as we go along. They lost my brainstem and cerebellum video when I was kind of previewing them. I'm like, we lost the cerebellum. So they told me they found it yesterday. It'll be ready today. It'll be ready in time for when we get to cerebellum. And of course the medulla. So these cranial nerves 1 through 12 are oriented from front to back, rostral to caudal. Okay. So this week, what's our bottom line? The medial surface of the brain, the gyri and sulci, a bit of the corpus callosum, and everything that we just highlighted and talked about in lecture as well. And this is a little bit newer, the basal view, but again, there's not too much on that. So take a look at that in the atlas and this material that I provide for you. And when lab opens up, see if you can go in there and recognize these with just some notes, and I think you'll be in shape. Again, next week, week 5, we're going to segue into the brainstem and looking at the 12 cranial nerves. Okay? Have a great day. See you soon.